Thanks very much, Martin, for the introduction. I'm delighted to be here today. And the other thing that's going to be a little bit different from the other talks is that I'm going to be focusing not so much on what we know about pain, but exactly how it is we know it. So I think it's really important that you hear in this series about how pain research happens and whether it's happening in an optimal fashion, because there are those of us that are starting to believe that there are problems in pain research, problems that go right down to the level of how pain research actually occurs. With that, I'll start, and by the way, that title isn't gratuitous. I'm going to defend that title. It's going to make perfect sense to you soon. But let's just start off a little bit about talking about pain and its importance as a clinical problem, because I don't believe either of the two speakers said this. At least I didn't see it on their slides. A few fun facts, if you will, about the problem of pain. The first one is this, and maybe this was mentioned actually, but pain turns out to be the #1 reason that people go to the doctor in the first place, or the chiropractor, or the acupuncturist. It's the #1 reason for seeking health care. It's the #1 presenting complaint. You go to your doctor, he says, "Why are you here?" And the answer to that question is, well, something hurts, more than any other complaint. Does anyone want to guess, by the way, what #2 is? And by the way, #3 is way, way down the list. So people go to their doctors, A, because something hurts, and B, because, any idea? Well, that would be because they have pain, right? But what are they complaining of to the doctor other than pain? Colds and fever are a good idea, but that's like #5 or 4. Say what? No, that would still be pain. I would still call that pain. Any other? Stress. That's a good idea, but no. You're never going to get it, so I'm gonna tell you now. The #2 reason is itch. Yeah. People go to the doctor when something hurts or when something itches, and everything else is way down the list. It's true. This is surprising. When they do surveys of people who have chronic and even fatal diseases like arthritis or cancer, and they ask them what are they afraid of, you would imagine that their #1 fear, their #1 concern would be, oh, I don't know, dying or not being able to move my hands anymore. But that's not their #1 fear. Survey after survey, people's #1 concern is whether their pain will be managed. That's what they're afraid of, even in the face of something like cancer or severe arthritis. And finally, generation after generation of medical students have been taught, and in some sense continue to be taught, not to worry too much about pain. Because, the idea is you take care of the disease and the pain will take care of itself and it'll go away. And besides, it said, pain can't kill you. Right? But in fact, pain can kill you. Now, it doesn't do it directly, but what chronic pain does is weaken people's immune systems so that they're more susceptible to developing cancers and infections that in fact do kill them. And it's been estimated that chronic pain takes, I think there was a recent survey in the UK, something on the order of like 4 to 6 years off your life. Okay? So pain, in fact, can kill you, and it's worth being dead serious about. We've made a lot of advances in pain research. The last lecture by Kathy Bushnell, she told you a lot about advances in brain imaging, and we now know about this pain matrix in the cortex, in the brain, where all the pain information is ultimately going. So we've made great strides there. This is very, very new. 10 years ago, we knew nothing about this at all. Something that Kathy probably didn't mention is that at the same time, there's been another revolution in biology, including in pain research, which is the molecular genetics revolution. So what you're looking at there is a screenshot of a database called the Pain Genes Database, which I made and maintain. And this simply lists all the genes that have been implicated in pain. Okay, now as you know, there are 22,000 genes, and many of them are probably playing some role in pain. And as the evidence comes into the scientific literature that a particular gene is involved in pain, I put it on the database along with some preliminary information about whether it increases pain or decreases pain. And I show you this simply to point out that if you go on this database today, you will find over 350 genes that have been implicated in pain, and that's a low estimate. Okay? At the end of the day, we're probably talking about thousands and thousands. We know about 350 plus as of today, but that's a lot more than we knew about 10 years ago. 10 years ago, 15 years ago, pain researchers were studying 20 molecules, literally 20 molecules. Now we're studying 350, and the number is growing every day. But here's the problem. Here's the dirty little secret of pain research. And by the way, I don't want to be too hard on pain researchers. This is not only the dirty little secret of pain research, this is the dirty little secret of all of neurology. Okay? The dirty little secret of everyone who is studying any disorder where the relevant biology is happening above the neck is that we haven't really gotten very far. So for mild to moderate pain, today we are using at essence, the very same substance as we were using 2,000 years ago, and Joe Schwartz told you all about this. Ultimately, we're still using willow bark. Now, it's gentler on your stomach than it used to be, and we've isolated the relevant chemical and blah, blah, blah. But at essence, it's still willow bark. It works the same way. For moderate to severe pain, we are still, as we were 2,000 years ago, using the opium poppy. And again, we can make it come out slower, and we can make it last longer, and we can, in some cases, mitigate some of the side effects. But ultimately, it's still the opium poppy. And the only thing really new in pain treatment in the last 100 years is the discovery that certain anti-epileptic drugs and certain antidepressants were also good for certain forms of chronic pain. And so people get gabapentin and pregabalin now, which were originally designed as anti-epileptics. And people get drugs like amitriptyline and duloxetine at very different doses than used for depression. But those are treatments for chronic pain too. But we shouldn't be too proud of ourselves here, because these discoveries are completely accidental. They had nothing to do with research. Right? It was simply that clinicians, doctors noticed, well, my epileptic patient may or may not be responding well to this anti-epileptic, but he told me that his back pain's better now. Right? So complete accidents. Okay? So people are starting to believe that something might be seriously wrong. So why haven't we cured pain yet? What's the problem? And it seems to me there's only a few options. Okay? One very serious thing is that pain research is massively underfunded compared to the scope of the problem. Okay? These data from 2009 from the major funding agency in the United States, the National Institutes of Health, and you can see how much funding is devoted towards cancer versus pain. Now, I will forgive you if you think that cancer is a bigger problem than pain. It certainly kills a lot more people than pain. Pain kills some people indirectly, but cancer kills a lot of people directly. But I would submit to you that the number of people affected by pain is actually bigger than the number of people affected by cancer. And people in pain have pain for a long, long time. And maybe cancer is a bigger problem than pain, but it's not 50 times the problem that pain is. And yet it's getting 50 times the funding. So that's problem number one. And by the way, things are no different in Canada. The second thing we've run into is we've run into a spate of some pretty bad luck. There have been a number of drugs that have been developed that actually look like they did kill pain, but they're not on the market. You can't buy them. You can't prescribe them. Why? Well, they had a side effect. They were toxic in some way. In fact, one famous drug, Vioxx, the toxicity didn't appear until after it was on the market, and then it was pulled off. To the extent to which we've gotten unlucky, this isn't my fault, right? This is not the fault of the pain researcher. Our job is to predict if the drug works. Toxicity is someone else's department entirely, right? I mean, it really is a completely different type of scientist. I know that sounds funny, like I'm ducking my responsibility. No, but it's not my responsibility. I don't know anything about toxicity. Other people do, right? It's possible that there are just species differences between the rats and mice that form the bulk of pain research and humans. I don't believe that's true, but it could be true. It's possible that the animal models we're using in those rats and mice just aren't very good. They don't accurately reflect the situation in humans. I do believe that's true, but that's not what I want to talk about today. What I want to talk about today is point number 5, which is complexity. Now, I mean this in 2 senses. Firstly, I mean pain is more complex than other things. The biology is more complex. In fact, while we're comparing to cancer, pain is way more complicated than cancer. Now, I know that sounds like a crazy thing to say, but it's true. Because in cancer, the biology, which is very, very complicated indeed, is all going on ultimately in one cell type, the tumor cell. Right? Whereas in pain, well, there are a lot of cells involved in pain. There's one in the dorsal ganglion, there's one in the spinal cord, there's one in the thalamus, there's one in the amygdala and in the cingulate and in the sensory 1 cortex and various areas of the midbrain. There are pain cells all over the brain. You have to understand each and every one of them. It's simply a harder nut to crack. But what I really want to talk about today isn't that kind of complexity. It's this kind of complexity. There are things that pain researchers aren't taking into account when they design their experiments that lead to individual differences, things like genetics and sex differences and social modulatory factors that can change the results of pain experiments. And by changing the results of pain experiments, we either come to the wrong conclusion or we come to conclusions that we generalize way more than we ought to. And so that's what I want to talk about today, and that, of course, explains my title. I'm going to talk about friends, social modulation. I'm going to talk about sex, as in sex differences. And I'm going to talk about your mother, as in genetics. All right? Okay. Just some musings a little bit about the concept of individual differences. The first person to note the importance of individual differences was no less than Sir William Osler, who, of course, as you know, was here at McGill for a while. And in 1892, he said, if it were not for the great variability among individuals, we could turn medicine from the art that it is into the science that it could be. Now, 1892 was a long time ago to appreciate that. He was really one of the first people who did. But, of course, everyone is talking about this now. And the phrase they usually use is personalized medicine. And the idea being, they talk a lot about this when they talk about drugs. Right now, drugs don't work for everyone. In fact, if a drug company has a drug that works on 35% of patients taking that drug, that's $1 billion blockbuster. But imagine how happy they would be and how much better society would be if there was a way to predict who would respond and who wouldn't respond. Okay? So lots of people are working on personalized medicine, including now pain researchers. So let's talk about the variability in pain. You can demonstrate this in a number of ways. You can demonstrate this in the laboratory. So what you're looking at here are data from an experiment that happened at the National Institutes of Health in Bethesda. I'm going to show you some data today, but every time I show you data, I'm going to put, hopefully, that little handy more pain, less pain arrow so you can tell which direction means what. In this experiment, 500 people got the identical pain stimulus, 49 degrees, delivered through that thermode to their forearm. Okay? The stimulus was exactly the same for everyone. And they were asked to rate the pain on a scale from 0 to 100, 0 being, I don't feel anything, and 100 being, and this is always the way they do it in pain experiments, 100 is worst pain imaginable. Okay? So no pain to worst pain imaginable. Look at the range of responses. There are people on the left end that thought that was about a 5, almost nothing at all. And there are people on the right end who gave that same stimulus a 95. Okay? As much variability as you can imagine, you can demonstrate in a laboratory experiment. And there's other data that I'm not showing you here that shows that this isn't just people using the scale differently. They're really having that different a perceptual experience. This is completely unexplained. Okay? This is probably more important, though. There are any number of insults, infections or diseases or traumas, that can produce the chronic pain syndromes shown in red. So some people with cancer have pain, some people don't. Some people having strokes develop shoulder pain or sometimes what's called post-stroke pain, and some people don't. Some people that get shingles go on to develop a horrible thing called post-herpetic neuralgia, where the pain of shingles, a pain that's so bad, my brother had shingles once, and he said if it had gone on for 3 more days, he would have shot himself for sure. Well, some people have that kind of pain for years, right? But most don't, et cetera, et cetera. And so the biggest question in pain research isn't, as you might imagine it would be, what causes chronic pain. The biggest question in pain research is really better asked as the following question. What is it about these poor people, the ones in the dark blue, right? Why do they go on to develop chronic pain syndromes when everyone else heals up just fine? What is it about them? And of course, maybe it's not them, maybe it's precisely what happened to them. But I think you can see how individual differences is the answer to the question, right? It's a question of variability. So how do you explain that variability? Well, you can explain it a number of ways, and the first way that pops into people's mind almost always is genetics. So let's talk about genetics for a little bit. I've been doing genetics for a long time, and the way I did genetics and continue to do genetics is by comparing the responses of inbred mouse strains. Now, inbred mouse strains are the result of brothers mating with sisters for enough generations that they turn into clones. Okay? So every 129p3 mouse in the world is exactly identical at every genetic locus, at every single nucleotide, as every other 129p3 mouse in the world. And every mouse that is 129p3 10 years ago, and every mouse that's going to be 129p3 10 years from now. They're all clones. Okay? And so it turns out by testing a bunch of these inbred strains at the same time for the same thing, you can figure out how much of the variability you see in something is genetic. And so we've done that for a whole bunch of pain tests. Okay? Now, I know you can't see the details here, but the details aren't important. All I want you to notice here is the following, that for every one of these graphs, each graph represents a different pain test. Some bars are high, some bars are low, and some bars are in the middle. What that means is that some of the mouse strains were sensitive, and some of them were resistant, and some of them were average. And you can actually quantify that, so overall, the heritability estimate you get is 46%, which is pretty close to 50%, which means that about half of the variability is genetic. If we find the genes that are responsible, we can explain half the variability. So that's a pretty tempting target because, after all, there's a large number of genes, 22,000, but 22,000 is a finite number. It's a big number, but we can work with it. In addition to there being differences between strains where one is more sensitive and one is less, you also run into qualitative strain differences every so often, and these are more worrisome. So I'm going to show you data from a test where you're looking at the amount of time it takes for an animal to hop away from a heat stimulus delivered from under a glass floor that it's standing on. Okay? So the heat goes into the paw, and first it's warm, and then it gets hot, and as soon as it's hot, it just hops away, and it's over. Okay? So here's what happens if you inject a chemical called CGRP. It doesn't matter what that is. It's calcitonin gene-related peptide, if anyone's curious, but it doesn't matter. You can see that there are some baseline measurements, and then you inject CGRP, and for a few minutes, the mice get more sensitive to pain. And then the CGRP wears away, metabolizes away, and they go right back to where they started. So that's all fine and good, and if you did an experiment like that, you would conclude that CGRP makes mice more sensitive to pain. Great, simple, right? And that would go into the literature, and everyone would know it, and that would be great, and people would use that finding and follow it up. But here's the problem. If you do the same experiment in another strain, CGRP doesn't do a thing. And it's not just a matter of the dose. Even if you try really high doses of CGRP, it still doesn't do a thing. So yeah, and it turns out that we did this experiment on like 14 different strains, and just as it happens, about half of them show a CGRP effect, and about half of them don't. So if you then try to answer the question, does CGRP affect pain, what's the answer? Well, it kind of does, but it doesn't. It does in some animals, but here's the problem. Very few people in the world ever do an experiment on more than one strain. So if the lab that did this experiment first happened to be using one of those strains, then a certain thing would come into the pain literature. CGRP increases sensitivity to pain. If the lab that did the experiment first happened to use that strain, what would go into the literature if a paper got published at all would be CGRP doesn't affect pain. What's the right answer? No one would ever know, right? And pain research is overwhelmingly done on one strain only. I'm going to give you one example of if you do take genetics seriously and try to come to the bottom of what gene it is, that you can find something, and you can find something in the mouse that tells you something about humans. So we got interested in a gene that makes a protein called the melanocortin-1 receptor, and it turns out that you can get your hands on a mutant mouse. The mutant mouse is shown over there. It's called the recessive yellow mutant. Okay? And it arose from that black strain that's labeled C57BL6, C57 black 6. Okay? So in a litter of C57 black 6 mice, one day arose a mouse with that beautiful tawny color. And it turns out that those two mouse strains are exactly identical, except that in the recessive yellow mutant, melanocortin-1 receptors don't work. They're made, but they don't work. Okay, they're non-functional. And it turns out that if you compare these two strains on a pain test, turns out if you compare them on a lot of pain tests, I'm only showing you one, you find that the mutant, the recessive yellow mouse, is less sensitive to pain. That's fine and good. Why am I telling you this? Because it turns out that there are people with mutations of the melanocortin-1 receptor walking among us, and they're called redheads. Okay? So redheads are essentially the exact equivalent of the recessive yellow mouse, in that they have variants that they have inherited. It turns out to take 2 in humans or more. And if you have 2 or more variants at this gene, you, just like the recessive yellow mutant, will have non-functional melanocortin-1 receptors. And one of the things that the melanocortin-1 receptor does is it turns your hair brown. That's what it's supposed to do. If it doesn't work, your hair fails to turn brown like it's supposed to, and you end up being a redhead. Well, the mouse experiment makes a very specific prediction about what we should see if we compared brunettes to redheads. So we did, and it's exactly the same. Okay? The pain test is a little different. This is electric shock tolerance in humans. So how much electrical shock can you stand before you say, I can't take any more? And redheads were less sensitive. They had higher tolerance, just like in the mouse. Okay? And there are now 5, 6, 7, 8, maybe 10 examples of where we found a gene, and when I say we, I don't only mean me, I mean scientists, but I've been responsible for a few of them, where we found a gene that affects pain in the mouse, and we've also found that it has essentially the same effect in humans. Okay? You really can go from mice to people in this respect. Okay, I want to go on to the second topic here, and this was set up in the introduction, sex differences in pain. Now, as Martin alluded, and a number of jokes have been made over the years, here's one of them, people believe that women must be more tolerant of pain because of childbirth. That turns out not to be true. Now, not every study that examines the question finds a difference. In fact, I'm working on a review paper just today, going over this literature. Not every paper finds a difference, but when they do find a difference, it's almost always, 99 times out of 100, it's in the same direction, which is women are more sensitive to pain, women are less tolerant of pain, and women are more able to tell one level of pain from another level of pain. Furthermore, it is certainly a true statement that there are some pain syndromes that have a female prevalence, some that have a male prevalence, and some that, as far as we can tell, are equally common in men and women. And there's a very famous table in the field listing them, and you can see that the pink list, pain syndromes that are more common in women, is a lot longer than the male list. And not only is it longer, not only are there more pain syndromes that women get more often than men, but some of the most common pain syndromes are in the pink list. And you put that all together, and the best estimate is that 70% of chronic pain patients are in fact women. Chronic pain is a disease of women. No one thinks of it that way, but the numbers bear out that conclusion. Now, what does this have to do with anything? Well, the problem is that fact, that epidemiological reality, is completely ignored by pain researchers. Okay? This is the modal pain sufferer, the most common single image that represents a pain sufferer. Okay? 55-year-old woman with a headache. Here's how we model that woman in the laboratory. Now, you're laughing because it's a rat, but that's not the problem. The problem isn't that it's a rat. The problem that it's a young male rat of a single strain standing in for a middle-aged woman with a complicated and diverse genetic background, right? We think that's good enough, but it's not good enough. How bad is the problem? Here's how bad it is. So I took a look at 10 years of papers published in the most prestigious journal in the field, which is called Pain. And I looked to see, I opened up every paper that used mice or rats and say, okay, what sex did you use and what sex did you report? And you can see overwhelmingly, 79% of studies are done in male rodents only. Few people specifically use females. A lot of times both were used, but no mention was made. Sometimes they didn't even report what sex they used. Overwhelmingly, pain researchers simply don't care that pain is a women's disease. They want to use male mice only. Why? I think they're afraid that the estrous cycle, which of course is the counterpart to the menstrual cycle in human women, they're worried that the estrous cycle in rats will somehow confound their experiment. We've shown this not to be true. But I think it's mostly just inertia. People do what they've always done, and it takes people a long time to change. You can get in serious trouble if you ignore sex differences in pain because, again, just like in genetics, there's two types. The quantitative sex difference, in this case, females being a little bit more sensitive to pain when you stick their tail in hot water. But that's not the worrisome part. The worrisome part are the qualitative sex differences. So I'll give you an example. There was a lot of interest for a while in developing a drug that was a combination of morphine, which of course you all know, and a drug called dextromethorphan. Now, the reason dextromethorphan was interesting to everyone was that dextromethorphan is an utterly safe drug. And we know it's an utterly safe drug because you've been taking dextromethorphan for decades. Does anyone know what dextromethorphan is the active ingredient in? Yeah, cough syrup, right? If cough syrup was dangerous, we would know it by now, right? And so the idea that you could put these two drugs together and get a better analgesic effect than just morphine alone was really attractive to people. And there was some scientific reason to believe it would work, because in rodent studies, they got data a lot that looked like that. If you compared morphine plus dextromethorphan to morphine alone, there was more analgesia, right? And that finding, these are our data, but believe me when I say that that finding was found in about 80 different studies. Over and over again, they found that finding. 80 different studies, and because it was found in 80 different studies, it led to a clinical trial of a drug called Morphodex, a one-to-one combination of morphine and dextromethorphan. That trial, of course, failed utterly. Morphodex was no more effective than morphine alone, and Morphodex obviously never came to market. Why did it fail? I don't know, but I will suggest one possible reason is this, that it turns out that dextromethorphan doesn't increase morphine analgesia in females. These are our data. Why didn't anyone know that? Because in the first 80 studies that tested morphine plus dextromethorphan, all 80 were done in male rats and mice. All 80. No one ever knew about the sex difference because no one ever looked, because no one ever used female rats or mice. That's the kind of trouble you can get in if you ignore this. And yet, as I just showed you, pain researchers ignore it all the time. Ultimately, those few of us that are specifically studying sex differences in pain have found enough differences in the operation of pain biology that I feel comfortable saying to you that when it comes to pain, males and females have different brains. The neural circuitry is different. The biology works differently in males and females. And of course, if that's true, then the direct prediction of that is that one of these days, some drug company somewhere is going to come up with a pill that literally will work as an analgesic in men but not women, or women but not men. That day is going to come. I don't know when, but it'll be kind of exciting when it does, because there's no example of that in medicine so far. And you can just imagine the marketing campaign, too. Right? It's funny, because when I first started talking to drug companies about these sorts of findings, I actually thought they would be excited about it. But actually, they kind of hated it, because I was telling them that they would have to, cut their market in half right from the very beginning. And I told them, yeah, but imagine the marketing campaign. Well, we'll see. One day. OK. We'll move over to a third topic, which is social modulation of pain. Now, this one is surprising even to me. But we've been doing experiments along these lines that we found quite by accident for about 6 or 7 years now. And the data that I'm about to show you are important because they really dig into the assumptions, the core assumptions of what's known as the biopsychosocial model. Now, the biopsychosocial model is the most accepted conceptualization of how ultimately we're going to explain health in general and pain in particular. And it simply posits that to understand something like pain, and to understand health or wellness, you have to understand biological factors, psychological factors, and social factors. Now, this is accepted by everyone now, okay? There aren't many hardcore reductionists left that don't believe that social and psychological factors have an important role to play. It is assumed that to study the biological factors, you want to use rodents. Why? Well, you just can't do experiments on people. Not really. You can stick them in scanners, and you can do correlational studies, but true experiments can't be done in humans. They wouldn't be ethical. They wouldn't be possible. They would be way too expensive, even if we could do them. So we do it in rats and mice. Fine. For psychological studies, I think everyone would agree that we would prefer to use humans, but it might be possible to model certain psychological states in animals, maybe. Okay? But I think most people would agree that if you want to understand social factors, you got to have people, right? Because mice and rats, what possible social life could they possibly have, right? Well, I'm here to tell you, they do have a social life. Now, it's not much, but it's something. They live in cages of 3 and 4 and 5. They, these days especially, have things to play with, to crawl under, to go through. They have dominance hierarchies. They cuddle each other for warmth. They compete with each other for access to food sometimes. They really do interact in a way that, probably isn't as good as your social life. Maybe is a little bit better than mine, but they have a social life. Right? I mean, if you were doing experiments on prisoners in solitary, they would have less of a social life than these laboratory mice, right? It's not correct, that assumption. There is social interactions between animals. They can be studied. Now you might say to yourself, yeah, but they have nothing to do with us. Well, I think that might not be true. What got us interested in this whole line of work was this chance finding. It involved an analysis of a large set of data collected on the 49-degree tail withdrawal test, which is simply you stick the animal's tail in hot water, you hold a stopwatch in the other hand, and first it's warm. You can try this in my laboratory one day. It'll be warm for a while, and then after a few seconds it becomes hot, and they just flick their tail out, and I stop the watch. Simple as that. What we found, much to our surprise, is an order of testing effect. So on average, the first mouse tested from a cage of 4 was less sensitive to pain than the second and the third and the fourth. Now, you have to understand how we do these experiments to make sense of this. We take the animal from its home cage, we take it over to the water bath, we test it, and then we put it back in the cage. We go to the next cage, take the first animal, the next cage, the next cage. And then we go back to the first cage and take the second animal. A minute or two has elapsed, right? In the data I'm about to show you, instead of putting mice back in the cage after we tested them, we took them over to a holding cage on the other side of the room. Okay? And when you do that, the effect goes away. Now there's only one difference between the orange group and the blue group. In the orange group, the critters are talking to each other, right? In the blue group, they're prevented from talking to each other. That's the only difference. Social communication must be affecting pain sensitivity. Now, I wish I could tell you why this is. I have no idea. We never followed this up because for some technical reasons, it was hard to work with this phenomenon. So we came up with another one that I want to tell you about. It has a terrible name. We call it socially mediated contagious pain hypersensitivity, SCPH. It involves another very simple and very old pain test with an unfortunate name. It's called the acetic acid writhing test. Everyone recoils in horror because of the name writhing and because of acetic acid, which of course, as you know, is vinegar. Let me say two things. First of all, it's incredibly diluted vinegar. And second of all, this is probably the mildest pain test in current use in the mouse. It gives them what is almost certainly a stomach ache for about a half hour, moderate pain at most. You can get rid of it with aspirin, okay? But it does have the advantage of producing a nice, easy-to-score behavior, which is called a writhe, which simply looks like that. They go like that. And you count up the number of times you see that over a half hour, and after 30 minutes, it's all gone. Now, under normal circumstances, you would inject a mouse with acetic acid. You would put it in a transparent cylinder to observe the writhes, and you would either count it up, or these days, of course, the video cameras record it, and we count it up later. Just one mouse to a cylinder. But in this particular paradigm, we added two more conditions. We added one condition where only one mouse is injected, but there are two mice there. And then we added a third, more interesting condition where there are two mice and both of them are injected. Now notice the difference between condition 3 and condition 2. In the both mice writhing condition, both mice are in pain, but they're also looking at another mouse in pain. Whereas in the other two conditions, there's only one mouse in pain, there's no pain to see. Okay? Does that make a difference? Well, of course it does, or I wouldn't be telling you this. Here's the data. Okay? Under the normal isolated condition, you get about 40 writhes. Here's what happens in the one mouse writhing condition, which is basically nothing. Okay? Still about 40 writhes. There's the effect. Okay? In the both writhing condition, now you get significantly more writhing, that is more pain, in both mice than you would get if they were tested by themselves. But here's the kicker. Only if the mice are cage mates. If you pull two mice from completely different cages that have never met each other before, that is, they're strangers, you don't see the effect at all, right? That's pretty crazy. And that reminded us of something, a phenomenon that's well known, and you're going to guess it because you learned about it last week. But this is empathy, right? It's a specific form of empathy called emotional contagion. Now, the thing about empathy that people don't understand is that empathy isn't one thing, it's a collection of things. And the most basic level of empathy is something called emotional contagion. And the most famous example of emotional contagion is what you're looking at there, which is contagious yawn. Right? So if my talk is boring you and you yawn, the person beside you might yawn even though they're not bored. Right? They're yawning simply because you're yawning. Right? It's contagious. Well, we think that pain sensitivity is contagious in exactly the same way in mice. Okay? Is it true in people too? Well, we actually just finished an experiment showing that it is true in people too. This time, we substituted mice with McGill undergraduates. I know, it's cheap. And we tested them for cold pressor pain. Simply put your non-dominant arm into ice water and ask for ratings of intensity and unpleasantness. How painful is that, and how bad do you think it is? And critically in this experiment, they were tested twice. Once by themselves, and once with another person who was either their friend that they brought in, or if they didn't have any friends and came in alone, we paired them up with another stranger that came in alone. And what we're interested in is not their pain sensitivity. We're interested in the difference in their pain sensitivity from when they were tested alone to when they were tested with the other person. And it's the same as in mice. If you're paired with a stranger, the difference, what you're looking at now is the difference between the 2 times they were tested. If they were paired with a stranger, there was no difference. In fact, if anything, they got a little bit analgesic, right, when tested with a stranger. But when they were tested with their friend, for both intensity and unpleasantness, the pain was worse. Now think about that for a second. That doesn't make any sense, right? If you go in and get tested for pain with your friend, it should make it better. It's your friend, it's social support, blah, blah, blah. Well, it doesn't work that way. It makes it worse. And that, of course, is precisely what we showed in the mouse. I've been talking so far about the influence of social behavior on pain, but we're also really interested in the opposite, in the effect of pain on social behavior, on social interactions. And so we've done a couple experiments along those lines. I want to tell you about one. And this could not be a more simple experiment. In fact, it's so simple it amazes me that no one ever thought of doing this before. You simply do this. You have 2 mice and they're in jails at the end of a corridor. Okay, literally jails, okay, vertical metal bars. Okay, so that the mouse in the middle, it can go and it can sniff and it can, interact socially with the 2 mice in the jails, but it can't lie on top of them or do anything like that, right? And one of them is in pain, and one of them isn't in pain, okay? And the question is, what does the free mouse that's free to roam, right? Where does that mouse go? Where does it spend its time? And we varied a whole lot of things. So we varied whether the mice were males or females. We didn't mix sexes in this experiment. It was complicated enough already, so they're either all 3 males or all 3 females. We mixed up whether they were cage mates or strangers because of what we learned last time. And we mixed up whether they were allowed physical contact or not. We could prevent physical contact by, instead of having a jail, we could have a plexiglass barrier. So now they could see the other mouse, but they can't touch them. Whereas with the jail bars, they can, right? And the question is, does any of this make a difference to where the free mouse spends its time? And the answer is that the free mouse simply could care less, spends equal amounts of time near the mouse in pain, near the mouse that's not in pain, or in the middle, except in one condition, which you can see second from the left there. If the mice were females, if they were cage mates, and if physical contact was allowed, the free mouse spent significantly more time near the mouse in pain. That is, they approach pain. They don't avoid pain. And again, if you think of it, that's nuts, right? Because the free mouse, what does it know about what's happened to the mice in the jails, right? They could have a disease. They could be contagious. It's very, very dangerous to go spend time with a mouse that's obviously in distress. And yet, the girls, if they're cage mates, go and do just that. Amazing. In a sense, this is more than just empathy. This is more than emotional contagion. This is starting to look like prosocial behavior. This is starting to look like nursing, right? And we sort of hinted at that in the discussion section of our paper, but we didn't do it very strongly. And because we didn't do it very strongly, we actually got scooped by someone who did this, I will admit, in a much more clever way. Okay? So this is the only piece of data I'm showing you that's not from our lab. No, I showed you some before, but anyway, mostly the only piece of data, not from our lab. This is from a lab at the University of Chicago, and it was very, very clever. So what they did, this is rats now, not mice. They had a situation where one rat was trapped in a plexiglass tube, and another rat was free and to inspect it. And it turned out that the door of the trap, it was possible to open it, but only from the outside. So the free rat could free the trapped rat. And eventually it would figure out how to do so after enough hourly sessions in this situation. And the question is, would the free rat free the trapped rat? Right? And they did a whole bunch of control experiments to show that there really was nothing in it for the free rat to do it. But yet, if you look at the black line over on the right, They did. With more and more training days, they got to a point where about 70% of the free rats would free the trap rat, and they would do it immediately. And they would do it instead of freeing chocolate in another container. They would free the trap rat first, and then they would go and open up the chocolate, and the 2 rats would share the chocolate. Amazing. It's really amazing. And they concluded that this was direct evidence for prosocial behavior. One interesting thing that I'm not showing you is, again, it was the female rats that were much more likely to do this and did it much faster than the male rats. But again, this is well in line with what we know about sex differences in human prosocial behavior, right? All right. Okay, I got 10 minutes left. I want to tell you one more story. Now, this is, in a sense, the most fascinating one of them all. This, by the way, is the story that's not yet published, so don't tell anyone. It's a story about social interactions affecting pain, but now cross-species, although not between birds and rats. It stems from the the observation of people in my lab, and people in my lab have been telling me this for years. They've been telling me that they see a watch pot never boils phenomenon, whereby when they're injecting mice with, let's say, acetic acid, and they should be showing pain behavior, they actually curiously don't show any pain behavior. It seems like the injection didn't work at all. And then the person injecting, once they finish injecting like the 8 mice, they'll leave the room. And of course, the video cameras capture what happens after that. And then when they look at the video, they say that, oh yeah, they definitely lick and writhe all right. It's just that they didn't start until we left the room, right? A human experimenter in the room appears to inhibit the pain behavior, okay? That's what they were telling me. Well, we decided to see if that was really true, okay? So, this is a pain test. It's not important what. It uses our new facial expression scale. This is why we were able to show this, because measuring pain by facial expression turns out to be more sensitive than the old way. So the total amount of pain you could have here is 2, so they have a little over 1, kind of a healthy, moderate pain. This is what happens when you inject the animals and you leave the room, as we usually do. Okay? So none refers to there's nobody in the room after the injection has been done. Okay? Here's what happens if instead of leaving the room, the person who injected or another person, it doesn't matter, simply sits down in a chair and reads a book for the next half hour. Okay? There's analgesia. It's true. What my people in the lab were telling me was absolutely correct. If there's a person in the room, it inhibits pain behavior. Okay? They appear to be in less pain. And here's the fun part. It's only the men. Okay? If the experimenters are female, we don't get any effect. And it's not a matter of physical body size, because I guarantee you that Ki is bigger than any of those men in terms of height. It doesn't appear to be that men are more physically imposing. It appears to be related to testosterone levels. And the reason we know that is that we did a little extra 2 experiments, which are pretty unscientific because there's only one subject. But I had my 11-year-old son come in on a couple occasions and read a book. Actually, of course, he didn't read a book. He played his Nintendo device. With the sound off, of course, in front of the mice. And you can see that he produces a middling effect, as would be expected from a prepubescent boy. We also got we got a senior, let's just say senior citizen member of our department to sit in the room and read a book. And again, you can see he produced an effect that was about halfway in between the men and the women. So it really does appear to be related to testosterone. The person doesn't have to be there. This is a purely olfactory effect. And the reason we know that is that we had people sleep in a t-shirt and then simply put the t-shirt in the room. So now there's no person in the room. There's only the t-shirt they slept in the night before. And again, t-shirts that were slept in by men produced the effect. T-shirts that were slept in by women didn't. And it has nothing to do with people necessarily. Because we get the same effect when we put bedding material from some other species in the room. So if you take the bedding that rats have lived on, male rats produces the effect. Bedding from female rats doesn't. If you take cat beds or dog beds, now those are all males, but in this case, instead of using males and females, what we varied was whether the cat or the dog was fixed or unfixed. If it was a fixed male cat or dog, no effect. If it was an unfixed male cat or dog, which were harder to find, they worked, they produced the effect, right? And so it appears to be male of any species, at least any potential predator species of the mouse. And this experiment, sealed it completely. You don't have to have anything in the room other than a vial of chemical. What chemical? That one. Androstadienone, which I've been practicing so I can say it properly for today. It's a hard word to say. Androstadienone turns out to be the pheromone that shows the biggest human sex difference. That is, the men in the audience are secreting from their armpit right now a whole lot more androstadienone than the women in the audience are. Now, there are many other pheromones that are coming out of our armpits, but the one with the biggest male-female sex difference is this one. And this is the famous one, too. This is the one that's been shown to affect female sexuality. If you use a high enough concentration of this chemical, you simply put a vial of the chemical in the room 1 meter away from the animals. You get the same analgesia as if there was a t-shirt or bedding or an actual live human being, right? So what does one conclude about this? Well, what I conclude is that mice, maybe all mammals, actually have an inborn fear of males. And how do they know it's a male? They know it's a male by smelling the androstadienone. Because all mammals, all male mammals, secrete exactly the same molecule. My androstadienone is exactly the same as a mouse's androstadienone. And every other mammal has exactly the same chemical structure. And so maybe when you smell this chemical, it tells you there's a male nearby. It doesn't tell you what species that male is. It just tells you there's a male nearby. But I've been thinking about this, and it strikes me that it almost doesn't matter what species it is. If it's a male mouse, that's potentially a problem, because you could get in a fight, right? You could get injured. If it's a male cat, well, then the cat could, eat me. Now, I know what you're thinking. You're thinking, well, a female cat can eat a mouse just as easily as a male cat can. That's absolutely true. But ask yourself, who's more likely to be hunting? The male cat or the female cat? It's almost certainly the male cat is more likely to be hunting. So it strikes me, it's actually not a bad idea to be afraid of males. And maybe, this is the way it happens. And what we're looking at here is fear-induced analgesia. And fear-induced analgesia is a very, very well-known phenomenon. So here are the conclusions. I could have more, but we'll boil it down to this. Pain in both humans and mice, okay? And there doesn't appear to be much of a difference. Now, I'm not saying there are no differences between humans and mice. And I'm not even saying there are no differences in humans and mice with regard to pain mechanisms. Obviously, pain mechanisms, and especially pain modulation in humans, is much more complicated and sophisticated than it is in mice. But the principles are the same. In both species, they're affected by genetic inheritance, and we're starting to find the genes. They're being affected by sex, and although we're starting to figure out, just how big a problem this can be, we have a long way to go before everyone does what they should be doing, which is do all studies in both male and female animals. And finally, and most surprisingly, in both humans and mice, pain can be powerfully affected by social factors. Factors that vary either in the life of the individual in question or social factors that have to do with the precise experiment that they happen to be in at the time. If we know this, and if we take this seriously, what's going to happen is this. Now, I appreciate that this might be a little bit too indirect for some of your liking, but believe me when I say that if anything's going to change in the treatment of pain, It's got to change first in how pain research is done. If we can convince people to do pain research better by taking account of these factors, right? Either controlling them or systematically varying them, not forgetting that they exist, then the findings from pain experiments that happen in mice and rats right now are going to be more accurate, less likely to be overgeneralizations, less likely to have missed something completely. And that eventually is going to work its way into the clinic, and more drugs are going to pass the clinical trials instead of failing the clinical trials. And that's the hope, and I think we're on our way. So with that, I want to thank everyone in my lab, past and present. I've shown you some of my collaborators in other labs over the years, and importantly, the funding sources that keep us going so that we can try to change how pain research is being done. And I'd like to thank you very much for your attention. Thanks.